Good morning. My name is Qunhua Li. I'm a faculty at Penn State University. So I'm a statistician. And my research is about statistical modeling for genomics data. So in particular, epigenomic data. And I work quite a bit on reproducibility. So then I was asked to give a tutorial. And then I think, as Ben said the other day, everything, if you add AI or LLM, immediately you become cooler. So recently we did play with LLM for some statistical data analysis, and in particular in the context of reproducibility. So I think I will also share that part of our insights or results with you guys. And as you guys all know, that the field of LLM advances really quickly, like weeks or maybe days. So if you find that what we did earlier this year is out of date, let us know. I'll be very happy to know your feedback. Okay, so I will first start with something I did earlier. So I think those work have been published. If you have seen them before, apologize for that. But I think it's probably a good message for students to realize the importance of reproducibility and the complication of measuring reproducibility in the scientific context, especially for high throughput data analysis. Okay, so I will start with this. So I think a few years ago, there were a lot of discussions about reproducibility crisis in scientific field. And then so Nature did a survey to see how people feel about this crisis. So as you can see that more than 50% of people felt there is a crisis. And then they break it down to different categories. So each of this is a factor that may contribute to irreproducible research. So I did a little bit of classification on this. So I classified this into 3 groups. So one is related to integrity, ethics, or review process. So you can see those ones with red arrows are those categories. But then what about the rest of them? So a lot of times people tend to think irreproducibility is about ethics or integrity, but actually, as you see, a lot of them are not. So the next category is about reporting. So if the reporting is not sufficient, then that made other people difficult to reproduce the research. Okay, so the blue arrows showed those categories. And okay, the third one is improper methodology. So basically that is if the practitioner does not use a proper statistical method or does not have the proper replication for the initial analysis or poor experimental design, then this could also lead to irreproducible research. Okay, so then for this talk, I will not focus on the first category. So we will focus more on the later 2. And in the past, when I talk about this, I focus on the statistical part, but then actually the LLM part is related to reporting. So I will talk about both the reporting issues and methodology, and then try to see what we can do to improve these 2 aspects. Okay, so here's what I'm going to talk about. So first, I will talk about how to assess reproducibility of findings from a high-throughput experiment and how to evaluate their reproducibility. And then another thing about reproducibility is the reproducibility of the entire experiment. So it's not about the finding, but the experiment as a whole, is it good enough? Is it the quality? So from the quality aspect. Okay, so can we, is there anything that made the standard method to assess reproducibility inappropriate for a certain type of experiments? And the third one is, if we can we use reproducibility as a criterion to help us design experiments such that we can have a reproducible experiment? Okay, and then the last bit is about the reproducibility of LLM, automated data analysis. So we all know that LLM has a strong ability to do code generation, and it can perform data analysis. But then the question is how reproducible the analysis are, and can we assess it or enforce the reproducibility? Okay, so I will try to be fast on the old work so that I guess people may be more interested in the LLM part, so that I can reserve some time for that. Okay, so I will just talk about some high-level ideas for the first three aspects. Okay, so the first one is about reproducibility of findings. So you probably know this very well-known paper, which has been cited for more than 13,000 times. So it has a very provocative title. It says why most published research findings are false. So when they say false, actually it often means not reproducible by another independent researcher. Okay, so then in this paper, so it actually is a very insightful paper. So I highlighted one sentence in the paper. So it says that the reason is because a lot of research findings, the claimed research findings are solely based on a single study assessed by formal statistical significance, typically for a p-value less than 0.05. Okay, so this sentence actually is pretty compact. It has several wisdom in it. So one is that if it's based on a single study, then that can be an issue. And another is if it's based on just some statistical significance score, then that could be another issue, because if you use one model for the same data, you use one model for the initial analysis, but using another model for the analysis, you can get different results. And also, this last bit is about the threshold. So when we use statistical significance, so there is a threshold. If different people choose different threshold, it can end up with different results. Okay, so basically this categorize some characteristics of findings from high-throughput experiments. So that is, we often would give the statistical significance, and then based on this score, we are going to threshold, and then choose the ones that are above threshold. So this is just an example from like a ChIP-seq data. So here are the signals. Each one is one signal. So this is a signal identifier, and this is significance score. And we threshold and choose the top ones. And so at the end, this is more or less like you rank and select. Okay. So this selection will be based on a threshold. As you can see, if I threshold, so what I'm plotting here are two replicates, and they are so they are ranked. So this is chopped a little bit. So you can see that if you threshold here versus here, then the reproducibility of your results, of your findings, will be different. Okay, so then that means if you do the threshold first and then assess reproducibility, for example, by the correlation, then the result would not necessarily reflect the reproducibility of the procedure, but it's confounded with the threshold. Okay, so then we actually did some work along this line. So what we did is instead of using a fixed threshold, what we do is we will say, okay, how about we view them as a whole, and then you can see that there are like two groups. So one is a more reproducible group, which are ranked higher, and there's a strong association between the two replicates. And then there is another group that's less reproducible, and they are ranked lower. They have a weak association between the two replicates. So then we just formulate this as a clustering problem. But this is a little bit different from the traditional clustering problem in the sense that now we have to deal with the ranks instead of the original data. So then we had a model called copular mixture model. So mixture model to capture the two categories, and copular to deal with ranks. So then at the end, we are able to calculate the posterior probability for a signal to belong to the irreproducible component. So that's this. And then we call that irreproducible discovery rate. So then this allows us to use this score to rank the candidate, and then in a way that's kind of independent from the threshold and the significance score of the values of the significance scores from the original analysis. And then the benefit is if you have, so let's say you use two different softwares, and then you can't compare the p-values from two different softwares because they are based on different models. But then if you use our procedure to see how reproducible the candidate ranking is across replicates valued by these two software, you can bypass that score and threshold issue. Okay, so here are some just real data examples. So this is a case with high reproducibility, and then you can see that after we calculate this score, and then we just plot the number of significant candidates at different scores, and then this y-axis is kind of like so we call that IDR. It's kind of like FDR, but for reproducibility. So you can see that even if we go down to relatively low list on the rank, we still have, we can control this irreproducible discovery rate. And then this is a case with low reproducibility, and you can see even with very few numbers of peaks, the irreproducibility goes up. So it was used in ENCODE to process the ChIP-6 data. And then this is an example to show when you use two peak calling methods, so one is max and the other is SPP. If you use the default threshold, you will find that the two peak callers don't agree that much in sense of the number of peaks identified at the same threshold. But then if you use IDR threshold, you will find that this disagreement actually is very much based on different threshold choice or incompatible threshold choice. Okay, so that's the first part, and then I will move on to the second part, that is to assess reproducibility of the whole experiment. Okay, so so the example I'm using here is Hi-C data. So I think on Wednesday you have seen Christina Leslie's talk. She talked a lot about Hi-C, so I think people are familiar with the structure of the data. So basically, Hi-C is to assess the interaction between two loci on the genome. And then once you get the experimental result, you can plot them, and they look like those triangles with this bottom of the triangle as the interaction between two loci that are very close to each other, and then the top will be too far away from each other. And as you can see that there is some kind of a gradient here from the diagonal to off diagonal. That is because when they are close, then there is a higher chance to have a random interaction. Okay, so this is called distance dependency effect. So then, so a while ago, my collaborator reached me to say, oh, okay, when we calculate the reproducibility of Hi-C experiments across the same cell type, we find that it could be even lower than across the different cell types. So that seems to be puzzling. And if you plot it, you will find that the within cell line concordance actually is actually higher than across cell line concordance. So why is that? Okay, so then if we go back to some very the statistics 101, this is a very simple question. So if you may see that people with well, old days, I guess, yellow middle finger may have a higher chance to have lung cancer. So then why is that? Is it the causal effect? Apparently there's not, right? In this case, you can immediately identify that smoking is the confounding factor that is associated with both. So, so the situation is the same here. So when we but it's just more subtle. So then when we consider two different cell types, both of them actually have this distance dependency effect. So this distance dependent effect made these two cell types have a high spurious correlation. And if you plot, you actually will find that you can see this is a plot against the distance against the interaction frequency. Then you can see that they have similar patterns. That's why that drives the spurious correlation. So then what we did is to correct this. So when we corrected this, we used a very simple idea. So we just stratify the Hi-C contact by the distance. Okay, so then this happened to be related to another old statistics. So it's called Cochrane mental hazard statistics, the Biometrics statistic. So when it was originally used, it's for some like clinical studies. So you have smoking lung cancer for different age groups. Then if you put them all together to analyze, that actually will hide some true correlation. So what you do is you just stratify by age. And then that's the same here. So we just stratify by distance. So we basically use we derive a formulation like CMH statistics. It's a little bit different because for CMH, the standard version, the signals so the stratifications are I think it's more discrete, and for this case, it's more continuous. So we made some changes, and then at the end we have this version. So basically, it's a weighted version of Pearson correlation, and it's pretty fast and easy to compute and easy to interpret. And then we call our statistic HiCRep, and then we did some we checked whether it works well or not by checking whether it can reflect the the relationship between cell types on the lineage. So then so this is statistic. So we call that SCC, so stratified correlation coefficient. And we compare with more traditional ones, and we found that our method can reconstruct the lineage relationship when we apply it to Hi-C data to compute the pairwise distance using our statistic. And then, so Bill Noble's group actually applied this metric and some other metric to compute the reproducibility, or as you can see, it's kind of like similarity metric. So apply this to single cell Hi-C data. So they found that our method is able to reconstruct the cell. So this data set is a cell cycle data. So what they did is to use our method to calculate the distance between two single cell, and then they use MDS to project, and then they find that our method can reconstruct the cell cycle relationship. So this from another way to show that this method is effective. Okay, so then the next thing we did is to use reproducibility to aid experimental design. So as you can see that if we can use the reproducibility as a goal, then we can check what kind of factors can affect the reproducibility of the experiment. So basically, so this is useful because often for high-throughput data or scientific studies, we don't know the ground truth. So we want to have experiments that are reproducible so that we can establish confidence. And then what we did here is, so what we did here is we built a regression model with reproducibility as the response, and then the experimental factors that you want to study, so like sequencing depth or maybe how many cells you need to go into your experiments or platforms with those things as the covariate. So one challenge here is how do you represent reproducibility as response? Okay, so as I alluded earlier, when we assess reproducibility, there's an issue about threshold. So this threshold can be a confounding factor. So what we want to do is to use a series of a threshold instead of a single threshold to avoid this kind of a confounding effect. So then what we did here is we use something that's kind of like a model in statistics that's called a cumulative regression model. So I think probably all of you know what a logistic regression model is. So logistic regression is, okay, so I have outcome and I just binarize that. So like if you have a threshold, you can say below threshold and above threshold. So if you have multiple thresholds, you actually at each threshold, you can do this. You can dichotomize to say, okay, below and above, and then for the next threshold, below and above. And then you will have a series of regression models. So together, they are the cumulative regression model. Okay, so but the traditional cumulative regression model is just for one dimension. And then here we have two replicates. So what we did is to extend it to two dimensions. So basically, we say we can like partition this result into this. So this is result into this kind of L shape thing. So then we will say, okay, below this L thing, that means they both are significant on two replicates, so they are reproducible. And if outside of this L, then it's not reproducible. And then we will change the threshold, and then we will get a series of those L-shaped things. And then we extend the model to a model that can take care of reproducibility on the y-axis. So that's so this is how our model looks like. As you can see here is the thing to take care of reproducibility, and then on the right side it's kind of like a standard regression model with some changes. Okay, so I will not go into the details on this. So we call that correspondence curve regression, and then we also extended this in two directions. So one is when reproducibility does not improve linearly. So if so in this model, it's kind of like a linear model, but then if it's not linear, if it's banded, so then we extend it to a segmented regression model. Okay, and then another extension is for high-throughput data, often we have missing data. So maybe some results, some measurements are observed in one replicate, but not in the other one. So then that they become like missing. So if you plot the rank scatter plot, you will see that you see the pattern like this. So those ones are the ones that are observed in only one replicate, but not in the other. So we extended that model such that we can model it using the regression, extended regression framework. Okay, so now I'm going to, so I think I have about 20 minutes left. So I will talk about the reproducibility of LLM generated data analysis. So I'll give a little story. So we started the project because like 1 year ago, I was the chair of our department's qualifying committee. Okay, so we need so for STAT students, they have a theory exam and also applied exam. Then for the applied exam, traditionally we do take-home exam, ask them to do some data analysis. But then because of the emergence of LLM, we are kind of concerned that people may cheat if they do the take-home exam. So then we were discussing this, and then one committee member in the faculty meeting just took the previous year's question, sent to ChatGPT, and then, like 30 seconds later, she got the answer. And then because she was on the previous year's committee, then she graded the question. She just took a quick look, and she said, well,
no, we can't do the take-home exam because ChatGPT can score maybe like 80 points, and that would be maybe above the average PhD first-year PhD students. Okay, so that's pretty scary, right? So then, yeah, I think we have the tools, and the tool is good. So 80% is not good enough for your analysis. If you want to use ChatGPT to do analysis, you can pass the qualifying exam, but you probably want to make your bar higher, right? So, so when you use the tool to do the data analysis, you get the results. You actually don't know if the results are right? So for data analysis, we don't know. So then the question is, can we trust the solution? And if you feel you can trust it, this is a fantastic tool, can just boost our productivity. So then apparently we know that LLM can hallucinate. You don't know when it does it. And when you see the result, if you run it twice, it may or may not give you the same result. So that is very concerning. So, so then if you look up online, then yes, so there are papers to discuss the consistency or sensitivity of LLM to prompt yeah, so not just for data analysis, but also other contexts. So now, this problem actually is not unique to LLM. So if we consider the data analysis problem done by human analysts, we have similar problems. So, so there is something called a garden of forking paths. So the data analysis process actually is sort of like this. So you go to a garden, and then there are paths. And if you go to different paths, you will end up at different places. So in this path, what we do is we have this workflow. And then in the workflow, we make choices, and we may choose different parameters, or maybe how we handle the outliers, or pre-process the data, or maybe, yeah, so there are, or model choice if there are multiple, then those all can affect the results. So, so like I think like back in between 2010 and maybe 2018 or so, so there were a lot of discussions about this reproducibility crisis. But then later, some other papers mentioned that, okay, maybe it's not just because people don't know statistics, don't follow ethics, or they don't document things well. In fact, if you use the same data set and you ask many researchers to analyze them, then they can come up with different answers because they have a different reasoning process, they have different choices. So then this actually also is a contributing factor to the variability of the analysis. Okay, so here are just some high impact papers, and then there are quite a few other papers in a variety of fields on this topic. So then what this tells us is something actually pretty obvious. So data science is beyond generating a single answer. Reproducible workflows are important. So you need to write down how you did it, and you need to write down in a good way, okay? So then what about LLM? Okay, can we trust it? So, so now one thing you can do, so we know that when LLM generates the result, well, if you ask it in a relatively good practice, then it will generate a plan. So that's reasoning. So sometimes you can see it. So for like a DeepSeek, it prints the reasoning directly. And some don't print the reasoning process, but it does the reasoning behind the scene. And then it also, you ask it to generate the code. Okay, so if you get the code, and when you want to decide if you want to trust it, you could review the code line by line. But then this defeats the purpose because we want to save time. And then this is time consuming. So, and also, if you are not very proficient in encoding them, you may not want to do this at all. So then, but the easier task is to review the workflow, okay? So if you review the workflow, then you can decide if the workflow is sound. And but then another issue is you want to know if the workflow is sufficient that it can be, the code it generates can be reproduced. Okay, so this is related to that kind of a documentation at the very beginning I talked about. Okay, so then what we did is we want to have a reproducible workflow such that as a human analyst, you can check the result, check the soundness. So what we did here is to follow this scientific process to define reproducibility. Okay, so we have this framework, we call that analyst-inspector framework. So if we think about how scientists do research, or maybe if you do some undergraduate, if you supervise some undergraduates to do research, you may feel you are confident with their coding ability, but you're not sure about the workflow they come up with. So what you could do is maybe you can pair undergraduate students together. So one student will generate the workflow in the code, and you ask another person to inspect, and then to generate, so if you want to know if the workflow is sufficient, so you generate the code from the workflow and see if this second person can produce the code that can give you the same result. Okay, so if it can generate the same result, then you feel the first person's workflow probably is sufficient. Okay, so this is the independent validation, right? Okay, so then once this, if you know that the workflow is reproducible, then you can check the soundness. You can see, oh, okay, if the workflow sounds reasonable. Okay, so that's the idea. And so we know that actually in scientific community, there are a lot of guidelines about how to write workflow, what you should include. But then there's not, so there are guidelines. So it's hard to measure, okay? But then with LLM, this actually becomes easier. So now we can have the original analyst as LLM and have this inspector as another LLM. And then we can just formulate what I said. So the first person, the first LLM generates the workflow and the code, and the second one will generate the code based only on the workflow from the first agent. And then if the second agent's code can generate functionally equivalent results as the first one, then we will call that workflow as reproducible. So this means that the workflow is sufficient to include all the necessary information. And if it has too much information, it can be distracting. But then if it can let the second agent generate the functionally equivalent code, then that means it doesn't have extra unnecessary information. So in statistics, we call this two concepts as sufficient and complete. So it's kind of nice to tap on some classical statistical concepts. Okay, so then the results can be affected by several factors. Okay, so one of them is the prompting strategy. So here we used some standard prompting strategies like chain of thought and the React. And then we also want to improve the prompting. So we created two strategies. So one is called, we call that reproducibility of thought, so RRT. So for this one, we just add a sentence. So we say, oh, okay, make sure another person can replicate the action by only looking at the workflow, and the action input reflects every step of the workflow. We just add this sentence to the prompt. And another is we created a reproducibility reflection. Okay, we call that R reflection. So that is, we will evaluate the reproducibility by the second agent. If the reproducibility fails, then we ask the analyst to revise or regenerate the solution. Okay, so then we evaluate our system using three large benchmark data. So they all have the statistical tasks. and they have different difficulty levels. So I have some examples here. So stack QA is the simplest. It's direct to say, oh, you compute what statistics, and then discovery bench is the hardest. So it gives you a question, and then the LLM needs to figure out what kind of statistical method it needs to use. Okay, so so that's the difficulty level, and then we tested 5 different LLMs. They are not the most expensive models, but they're pretty good. And then we have several different inspectors. So the first thing we want to see is, okay, so we assess the reproducibility. So this is an actual step. Is it true that the reproducible solutions are more accurate than the ones that are not reproducible? So we did this plot, and it showed that in all 3 data sets, if something is scored as reproducible, then it has a higher accuracy than the irreproducible ones. And then this one compared several things. So one is to compare the 2 reproducibility enhancing prompts, so the 2 red ones, with the other, the baseline. And you can see that the 2 red ones would generate more reproducible solutions. And this is a little bit less visible, but the accuracy in general is higher. But it also depends on the data set. Okay, and I guess some of you are interested to know which LLMs are the best. So in our test, the O3 mini is the best, but it has a better reasoning than the other models. So that's not surprising. So it's more reproducible and more accurate. And interestingly, Cloud has a high accuracy, but the reproducibility is relatively low. And also, as you can see, we have 3 data sets with different difficulty levels. So the y-axis are on different scales. So the discovery bench, the hardest test, the questions with the most ambiguity has the lowest accuracy and reproducibility, and then the most straightforward request stack QA has the highest in both. Okay, so then we also look into the cost of irreproducibility in the failed workflows. So we found that majority have the issue with insufficient workflow, so they miss some steps, and then followed by misspecification. So that means the steps are included, but they, for some reason, the code actually didn't implement it correctly. And then also there are some over-specification, so it includes some unnecessary steps and confuse the agent to generate code. And then when we have the inspector, so that's in the R reflection prompt, it can reduce the proportion of all the 3 types of irreproducibility significantly. Okay, so we have 3 different inspector agents, and we checked them. They all are pretty, so our results are pretty consistent across these 3 inspectors. And interestingly, we actually found that Cloud is a little bit, so it has a favoritism to its own analysis. But then for the other 2, the O3 mini and the GPT-4o, we don't observe that. Okay, so then one thing I want to say is, so this framework, as you can see, it's pretty simple, and it's pretty generic. So basically, you can apply that to many different scenarios. So we just kind of add an inspector, sort of like how we do that in scientific community, to have independent validation. Okay, so yeah, so so this will be my last slide. I don't want to block you from lunch. So here are just some takeaways from based on my experience with reproducibility. So I think reproducibility is an overloaded word, and itself is indeed pretty complicated. So lack of reproducibility can be due to many different reasons. And then we often need to consider this case by case. So how to measure reproducibility, how to properly measure it, depends on the context. So like what is the problem at hand, and what is the data structure. So like high-C data, they have this dependent structure. And also, we need to be aware that sometimes the variation is inevitable. Okay, so it doesn't necessarily mean that it's irreproducible, but this means that we want to do the best practice to have the right documentation. And LLM actually can help automate this and enforce this. It actually makes the documentation easier. And with the platform I showed, it actually can assess this reproducibility by agent, so you can do this pretty quickly. Yeah, so last, I think everyone knows this, but sometimes we forget. So we want to enforce reproducibility, but it's not, so enforcing reproducibility can enhance accuracy, but they are not the same thing. So if things are reproducible, it doesn't mean that it's necessarily right. So sometimes we need to avoid making the same mistake or systematic mistakes, and they are reproducible, but they actually are not accurate. So like in the LLM case, we actually need to be a little bit careful. If your agents have the same training, then it could generate a similar right answer, or it thinks it's reproducible, but it's actually incorrect. Okay. Okay, so with that, I just want to acknowledge people who contributed to this work, my collaborators, and funding. So if you have questions, I would be happy to answer them.